Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. So, this is lecture number 15, where we will talk about isoquinoline alkaloids, biosynthesis and tissue localization. So, isoquinoline is general group. So, here mostly we will be dealing with the one particular type of isoquinoline alkaloids, which is called benzyl isoquinoline alkaloids. Let us go to the concepts which are going to be covered. First, I will talk about opium poppy, then I will talk about isoquinoline alkaloids and their classification. Next, I will talk about the pharmaceutical properties of some of the benzyl isoquinoline alkaloids, and next we will talk about the biosynthetic route, what are the enzymes, what are the steps, because this is very important. Once we know this, then it will be possible for us to do the genetic manipulation, which will come under metabolic engineering in the subsequent classes. And then if time permits, we will talk about cell and tissue localization of these alkaloids, otherwise we will cover it in the next class. Opium poppy. So, opium poppy, this is basically an opium poppy plant, and what is showing here is basically the poppy flower as and the mature fruit, which is botanically called capsule. If you make a sharp incision with a blade, and then you will see this milky sap or droplets are coming out, which are nothing but these are called latex, and these are the rich source of morphine and related alkaloids such as codeine. So, that means normally the capsule needs to be wounded, then only the latex will be exudated. So, this latex can be collected and later tested for the presence of these alkaloids, and when this latex is allowed to dry, it forms a hard brown substance, which is called opium. So, isoquinoline alkaloids derive from the condensation of phenylethylamine derivatives with a phenylacetaldehyde derivatives. So, both the motifs basically are come from the same precursor, which are either phenylalanine or tyrosine, and the prototype alkaloid of this is papaverine, which is found in this Papaveraceae family, that is the poppy family, and isoquinoline alkaloids are found not only in Papaveraceae family, but also in Barbaridaceae family as well as in Ranunculaceae family. And this is a very large class of medicinally active alkaloids, and that is why we are studying this. So, here there is an overview of the biosynthetic pathway for 4 groups of plant isoquinoline alkaloids. So, first one is the benzyl isoquinoline alkaloid. So, it contains an additional benzene ring. So, that is why it is called benzyl isoquinoline, which basically we will discuss because lot of work has been done on this aspect and the enzymes and genes have been worked out. So, it is possible to do genetic manipulation, and in fact, there are very nice success reported with this pathway. So, we will study that. So, other one is the phenylethyl isoquinoline alkaloids. So, this is one group, and the example of this is phenylethyl isoquinoline alkaloid is colchicine, and for your information, colchicine biosynthesis has been elucidated only few years back, and that published in a very high pack journal. So, we will cover the colchicine biosynthesis a little bit later once I finish the metabolic engineering application, and then before ending the isoquinoline group I will cover the colchicine. The third one is the ipecac alkaloid. So, the example is emitin, and emitin has a lot of pharmaceutical applications, and we will not cover that, but what I would like to mention here that for benzyl isoquinoline alkaloids, scientists put their effort for the last 30-40 years and they elucidated the pathway, but for ipecac alkaloid it was not the case. So, there was some problem to work with the system. One most important point was that the cell culture system, because isoquinoline or benzyl isoquinoline alkaloids can be induced in the cell culture, and cell culture is a nice system where the pathway can be induced and subsequently the enzymes can be characterized, purified can characterized, and subsequently the genes can also be isolated either by traditional approach or by modern PCR based approaches. Okay. For EPICAC that was not the case, only the breakthrough happened after the discovery of after the invention, I should say, after the invention of new molecular tools which is called transcriptomics. So, using the transcriptomics tool, now scientists have elucidated the emetin biosynthetic pathway in EPICAC. So, we are not going to cover that because we have lot of other things to cover. So, therefore, we will not put much attention, and amyloid alkaloids is another group of alkaloids, and the example is lycodine, galanthamine. Galanthamine is basically a very strong acetylcholine esterase inhibitor. So, it is used as a drug. And the examples of benzyl isoquinoline are morphine, reticulin, scopolamine, which we will discuss soon. Now, these are the structures of left side, these are the structures of different alkaloids, and in the right side what I have only showing, what I am showing here is basically the major alkaloids of pharmaceutical importance, one from the morphinan group, that is codeine and morphine, and another one is from the benzophenylthranidine group. So, which is sanguinarine, and the third one is from the berberine alkaloid, which is the example is berberine, and fourth one is the protopine alkaloid, the example is protopine. Okay. Next, what about the pharmaceutical importance? So, pharmaceutical importance, although I have spoken in the previous, in one of the previous classes, but just to brief up, for example, morphine is a very strong analgesic. Codeine is also a strong analgesic, plus this has anti-tussive properties, that means it is a cough suppressant. Similarly, other ones such example papaverine. Papaverine is also used as a vasodilator, vasodilator. Thibane is also an analgesic, and reticulin is basically a hair growth stimulator. And there are other products, for example, some are the microbial biotransform products. Some of the products formed from microbial biotransformation, and we will also list a few products which form from chemical transformation. So, microbial biotransform products are hydromorphone. So, morphine can be transformed into hydromorphone, which is also a mild analgesic. Similarly, codeine can also be transformed into hydrocodone, which is also used as mild analgesic in the pharmaceutical preparation. Similarly, there are chemical transformation products like oxycodone, and buprenorphine, which are also used in different pharmaceutical preparations. This is N-oxycodone. And few more, let me find out. Noscapine, another alkaloid. Noscapine is also antitussive. Papaverine, also vasodilator, also it is a muscle relaxant. And papaverine is also used as antispasmodic. Other examples are ipecac. So, for ipecac, this amitin is basically extracted from ipecac. So, that is used as a poison antidote. Emetine. And curare, curare is basically a muscle relaxant drug. Another example is chelidonine. That is also a mild analgesic, and berberine this is antimicrobial, and of course, the one which I am going to cover, which is the last one, is the colchicine. Colchicine has many pharmaceutical application, including as antineoplastic agents. So, you see, so this particular group has a lot of applications in the pharmaceutical industries. So, let us go to the next slide. So, now I will not discuss that much details of this. So, this is basically talking about the structural subgroups. So, the blue designates the each molecule originating from the tetrahydroisoquinoline moieties, and the red designated the part of each molecule originated from the benzoylic moieties. So, these are the examples. So, this is bis benzyl isoquinoline alkaloid, and this is particularly the structure of morphine and alkaloids. This is just for your information. Now, we will come to the biosynthetic route. So, to start with, as I said before, the amino acids are the precursors of this alkaloid. So, tyrosine Tyrosine will make tyramine. Tyrosine can also make dopa, and from dopa a dopamine will be formed. I will join it, and from tyramine in order to make a nice scheme, I need to erase the tyramine from here and add it here. And from tyramine it produces 4-hydroxyphenyl acetaldehyde. So, let us now join this group. So, tyrosine will make tyramine, tyrosine will also make dopa, from dopa to dopamine, tyramine to this. So, the enzyme responsible for this is I will put it here. One is this tyrosine decarboxylase, which is TYDC, which converts tyrosine to tyramine, and by the process it releases a molecule of carbon dioxide, and then tyramine through multiple steps it converted into 4-hydroxyphenyl acetaldehyde. Again in the other side, tyrosine is converted to dopa by a phenol oxidase, and and dopa to dopamine again by a tyrosine decarboxylase. Now, these two molecules, that is 4-hydroxyphenyl acetaldehyde and the dopamine, will join and forms a compound called norcocloudin. So, norcocloudin is the starting point of isoquinoline alkaloid, particularly benzyl isoquinoline alkaloids, and then these subsequently converted into cocloudin. Cocloudin, and then cocloudin to N-methyl cocloudin, methyl N-methyl cocloudin, and then N-methyl cocloudin is again the branching point of several routes. So, one goes in this direction and forms compounds such as bis-benzyl isoquinolines, bis-benzyl isoquinolines. Leading to formation of different compounds. So, this is not our point of discussion, that is why I am not going to discuss this, but what we will see, the main pathway which moves further down and makes S 3 prime hydroxy N methyl co-clouding. Okay. It's a big name, don't get daunted. So, it is no problem, you do not have to remember anything, and these next steps ending up in the formation of reticulin. So, S reticulin. Now, let us put the enzymes or whatever is known. So, this is called norcocloudin synthase NCS, which joins 4-hydroxyphenyl acetaldehyde with the dopamine. The next one, norcocloudin to co-cloudin, is basically 6-O-methyl transferase, 6-OMT. The next one is the co-cloudin CNMT, co-cloudin N methyl transferase. Okay. And the next one, from N methyl transferase to 3-hydroxy N methyl co-cloudin, this is basically a cytochrome P450 enzyme CYP80B1, this is its name. Okay. And the next one is basically the 4 prime O methyl transferase. So, this makes reticulin. So, reticulin is the branching point. So, this is basically the early step of benzyl isoquinoline alkaloid biosynthesis, starting from the precursor tyrosine, and it makes norcocloudin, co-cloudin, N methyl clocloudin, and this requires lot of methyl transferase, that is why 6-OMT, CNMT, and then 4 prime OMT, and in between there is a hydroxyl group addition, that is why CYP80B1 is there. So, when I will show you the structure, so this will be clear to you, and so now let us move into the next step. So, next steps we will start with reticulin. Reticulin will convert it into scoularin or this scoularin, I will use a different color. Scoularin, scoularin, and from this different pathway emerges. One leads to the formation of berberine alkaloids, another one leads to the formation of protopine alkaloids, another one to the benzophenanthridine alkaloids. So, let me show you that. The first one is the berberine alkaloids, example is berberine. Second one is the protopine alkaloids, and the third one is the benzophenanthridine alkaloids. Okay, the examples are sanguinarine, protopin is protopin and here is berberine. Copticin is another example which will also come under berberine. Okay, so, and the most important point is this, the enzyme which converts this to this is called, first one is called BBE, berberine bridge enzyme. So, berberine bridge enzyme is very important which convert S reticulin to berberine. Now, the other to the scoularin and then scoularin moves in different direction. The other pathway from reticulin, it moves in another direction and through multiple steps we see the formation of salutaridin, salutaridinol. I will discuss detailed steps in one of the classes next. Okay. Then this forms thebaine, 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 and from thebaine its one route goes to just I put the arrow first. And the other route is oripavin and then morphin to okay. So, the thebaine one is the neopenone, then codinone, then codeine and then morphine, whereas the other route from neopenone, it makes oripavin and from oripavin it makes morphinone. Oh, sorry, I have to use morphinone, and from morphinone it makes morphine. So, there are two routes we will talk about because of shortage of time. I am not discussing these details here and not the enzymes responsible for these steps. So, in the next class we will talk details of this pathway. I will talk details of this pathway, the enzymes and why there are two different route exist. We will talk about that. And so, this is basically in brief that from reticulin there are two major pathways originate, and not only that there are other multiple pathways also originate which we will show, and this leads to the formation of the morphine and alkaloids, which is basically in the right side what I have circled with yellow. And the other one on the left side. So, we will discuss here just to tell you that the alkaloids are accumulated in the latex So, these alkaloids are accumulated in the green latex, that is why in the background it is showing green, whereas the sanguinarine accumulated in the root. Okay. So, in the plant. So, sanguinarine is also produced by opium poppy, and so opium poppy produces a range of alkaloids including morphine, codeine, thibane, sanguinarine, protopine, everything. And the reticulin is the point here is this reticulin. So, the reticulin, the S reticulin is the starting point. In one way it goes to scoularin, and the other way it goes to R reticulin, and then it forms the downstream pathway. So, which we will again discuss in the next class. So, this is in brief of the some of the plant species which are worked, which scientists explored for the pathway discovery. One is important Papaver somniferum, that is opium poppy, and from here different alkaloids are formed. Similarly, Eschscholzia californica, that is called California poppy. So, that is a good source of sanguinarine. So, that has also explored. This is Archibon mexicana. This is Coptis japonica, that is a good source of berberine and copticin. Columbamine, when we will talk about this pathway, we will mention this. Similarly, Thalictrum and berberis, these are the important medicinal plants and which are a rich source of these alkaloids. So, reticulin is important. So, that is why I put it here, and in the next I will end the class with this slide. So, reticulin, the chemical chameleon or chemical chameleon, whatever you say. So, it is a lizard which can change its color. So, basically the structure reticulin with modifications, it formed different group of different type of alkaloids. So, for example, morphinan alkaloids, papaverine, berberine alkaloids, then benzyne threonine alkaloids, which is here. So, that means the reticulin is basically the starting point, and here I am showing you the few photographs of the plant, like protopine is produced by the Fumaria, this is Fumaria officinalis, and this is the sanguinaria which is called bloodroot. So, this is the rhizome part where mostly the alkaloid accumulates, and this is the aerial part, and this is the choroidalis cava, and this is berberis, okay, and this is basically the there is another plant, and this is basically the Papaver somniferum, this is a poppy. Okay. So, with this I end the class here, and thank you very much.